Hello students. Now we will see a little more detail of recombinant DNA. In the previous video we had just seen a general description of how recombinant DNA is formed. So let's see the construction of an artificial recombinant DNA molecule on an artificial scale, how a recombinant DNA was formed. So Stanley Cohen and Herbert Boyer. They too did this work of forming an artificial recombinant DNA molecule. And where did they do this work in? What did they use for doing their work? They used the bacterium Salmonella typhimurium. So what they did, they first identified the DNA with the desired genes. So what did I tell you? What is the desired gene? That means the gene of interest. So they first identified the gene of interest which had to be injected into the host organism. So first they identified that. Then what did they do? They cut the DNA. So they identified the desired gene, whatever foreign gene was there, whatever that has to be incorporated, they first identified that and they cut that DNA. So how is the cutting done? The cutting is done with the help of DNA scissors, and I told you what are the DNA, which is the enzyme that is involved, restriction endonuclease. So this is called the DNA scissor. So this is the restriction enzyme, or you can say restriction endonuclease. So what does it do? It can cut DNA at specific location, and we get the desired DNA piece which has to be incorporated into the new host. Then what is done? We obtain, so that first the DNA with the desired gene is identified, then the DNA is cut, and then next step is what? They are added to the plasmid DNA. So this foreign gene is added to the plasmid DNA. So now we are having this recombinant DNA. So what does this plasmid DNA do? It acts as a vector. This plasmid DNA acts as a vector. So what is a vector? Vector is a vehicle of DNA molecule which help to transfer the cut piece of DNA. We have, what is our main aim? We have to transfer this into the host organism. So this plasmid DNA, it is acting as a vehicle which is transferring this into the host organism. So this plasmid DNA act as vectors. So what is plasmid DNA? It is a circular DNA which is capable of replicating independently. So when we are putting this into the host, it can be able to replicate. So we can obtain the progeny also with the desired characters. So formation, this is what is called formation recombinant DNA. What is formed? Recombinant DNA. So the three basic steps that are involved. Now what we are forming a new organism. So we call that new organism that is formed, that is genetically modified organism. That is, we are modifying that organism with our gene of interest. So that is called GMO, or genetically modified organism, or we can say gene cloning. After the organism is formed, we are obtaining copies of that. Okay, so the three basic steps that we can say is, first is identification of DNA with the desirable gene. So as I told you earlier, this DNA you are identifying. Second is introduction of identified DNA into the host. So how is it done? It is done with the help of vector. What is the vector? Plasmid DNA. So first we have to identify the DNA that is incorporated into plasmid DNA. Then plasmid DNA will help introduce this into the host. Now after it is introduced into the host, we have to see whether that plasmid DNA which is in or the recombinant DNA which is introduced into the host, is it doing its work? So the third step is maintenance of introduced DNA. Okay, so that is the third step, that is maintenance of introduced DNA in the host. And we have to see whether it is working there, whether the progeny is also getting that same character. So these are the three basic steps that are involved in gene cloning or in genetically modified organism.